So Andrew, thanks so much for coming. We're just super excited to hear about your work. Many of us know about Genomic SEM, but really want to get a more in-depth knowledge. So very happy to have you here and welcome.
Thank you. Yeah, it's a real pleasure to be speaking to this group in particular. And today I'm going to be talking about Genomic SEM overall, but I really want to focus the second half of my talk on the application of Genomic SEM to psychiatric traits. And I know a number of you have heard me talk about Genomic SEM before, so I'll try to keep this first part relatively brief and more just a refresher. But there is a new package update within the last couple months that I've put on the GitHub for stratified Genomic SEM. So hopefully that'll be kind of a new thing for some people here. So just to start giving an overview of Genomic SEM and kind of the motivation for developing this package. I'm just showing here an old plot from Kendler et al. 2012 showing GWAS hits on the y-axis and different traits on the x-axis. And what this is demonstrating is something that's well known to everyone here, that as the sample sizes have increased for different GWAS studies, that the number of hits that we've identified has increased in a corresponding way pretty rapidly, where we're now identifying hundreds of different genetic variants underlying complex traits that are of interest to people. And so for me, as someone who's interested in psychiatric outcomes like schizophrenia and depression, if we look at these two Manhattan plots from some of the more recent efforts from these groups, these traits are so polygenic that if you're trying to figure out what actually is shared across these two traits, it's not just a matter of identifying 5 or 10 overlapping genes or loci that are shared across these Manhattan plots. And it really required people to think about how we could actually quantify the level of genetic overlap. And that was done really beautifully in 2015 when the group from the Broad introduced LD score regression, and more specifically bivariate LD score regression for examining shared genetic overlap across traits, which allows you to estimate genetic correlations between samples with varying degrees of sample overlap using what is often publicly available GWAS summary data. And when people apply Genomic SEM, like they did in the Brainstorm Consortium paper in 2018, you can produce these genetic heat maps across different sets of traits, where on the left, you've got psychiatric phenotypes, and on the right, across a wider range of kind of brain disorders and behavioral cognitive phenotypes, where the darker shading indicates higher levels of genetic overlap. And unsurprisingly, we see that there's certain clusters of traits within these heat maps where, for example, psychiatric disorders tend to show a pretty strong level of genetic overlap, which is something we might expect based on high levels of comorbidity that we observe among psychiatric disorders, while at the same time, some genetic correlations were maybe particularly or even surprisingly high, such as genetic correlations between bipolar disorder and schizophrenia in the range of 0.6 or 0.8, as you can kind of see indicated by this particularly dark blue box between those two disorders. So when these sorts of papers were coming out, I was in the midst of running twin models and helping navigate and manage a twin study down at UT Austin, and running different multivariate models of twin correlation matrices. And so seeing these correlation matrices coming out based on genomic patterns of convergence, there seemed to be this kind of strong need to really develop and apply multivariate methods to actually model these constellations of genetic overlap. And so that led us to develop and publish this paper that introduced genomic structural equation modeling in 2019 in Nature Human Behavior, which is kind of broadly speaking a framework for modeling patterns of genetic associations across wide constellations of traits. So in general, Genomic SEM uses a two-stage estimation procedure, where in stage 1, you estimate the genetic covariance matrix and associated sampling covariance matrix of standard errors and their codependencies. We use LD score regression within the context of the package, but you could hypothetically use any sort of genetic covariance matrix, such as you might get from Gremel. And then in stage 2, we actually fit the structural equation model to the matrices that are estimated from stage 1. So just to kind of show you visually what those stages look like. So in stage 1, we'd create this genetic covariance matrix, or what we call S, that has the heritabilities estimated from the genome-wide data, on the diagonal, and those genetic covariances or coheritabilities on the off diagonal. And then critically, we're also estimating this sampling covariance matrix V that contains the squared standard errors on the diagonal and the sampling dependencies on the off diagonal, which indexes the dependencies between estimation errors. And that's what allows us to apply Genomic SEM for samples that have different levels of sample overlap. And that doesn't need to be known. That is directly estimated from the data using a block jackknife procedure. But again, if you have different sets of summary statistics and you're worried about some level of overlapping controls, this sampling covariance matrix is going to allow you to produce unbiased standard errors even in the presence of that sample overlap. And then in stage 2, you take those two matrices and feed it into the Genomic SEM R package and specify some sort of model, like this genetic multiple regression model, in which we have schizophrenia and bipolar disorder as correlated predictors of educational attainment. And then parameters are estimated that fit the observed genetic covariance matrix as close as possible. And since this is a fully saturated model, the model parameters are simply a transformation of the observed matrix. One thing that I like to highlight when I'm talking about this, and I know that this kind of first part of this phrase is not applicable to this group, but even if you are not interested in genetics, I think Genomic SEM offers some kind of valuable tools because it allows you to look at systems of relationships across a wide array of rare traits that could not be measured in the same sample. And so if we think about just a case example of what that might look like, let's say that as someone interested in clinical phenotypes, you have a real interest in the relationships between schizophrenia and bipolar disorder. You've read the research literature and see a lot of convergence across different risk factors for these disorders. You have a pretty good understanding that these phenotypically can often look pretty similar. And you also notice pretty similar kind of age of onset distributions for these two disorders. So in that case, you might be interested in looking and quantifying in a sort of cross-leg panel like this, the relationships between both early and late onset versions of schizophrenia and bipolar disorder. The issue is that these two disorders are actually not, you can't diagnose them together. In the DSM, these would be rule outs of one another. So you couldn't, in a phenotypic sample, actually observe these two disorders within the same individual. And so you wouldn't be able to estimate this part of the model using phenotypic data. And of course, you also can't observe both early and late onset versions of a disorder within the same person. So you couldn't look at this part of the model. But with Genomic SEM, you can actually stratify the GWAS summary statistics by early and late onset versions of these disorders, and you could actually fit this sort of model and actually start to test the sorts of relationships that we've been left to really just hypothesize about and make qualitative conclusions around based on convergence from different separate univariate studies. At this point, stratified genomic SEM can be used to look at convergence at 3 main levels of analysis, and I just want to walk through those. So at the genome-wide level, this is actually just a recapitulation of that multiple regression model I showed earlier as an example of stage 2 estimation, where we're just looking at the system of relationships between schizophrenia, bipolar, and educational attainment. But this is just to highlight that for people who are interested in sort of mediation type hypotheses, that within Genomic SEM, you can kind of quantify things like a total indirect and direct effect between different disorders. Within Genomic SEM, you can also get standard model fit indices like AIC, model chi-square, CFI, and SRMR, and that allows you to go through kind of classic model fitting procedures to try and decide between different competing models and decide what kind of patterns of relationships best fit the data. So in that original paper, we looked at a series of models fit to the covariance matrix estimated across these 12 neuroticism items. And what we found is that a common factor model fit the data pretty well, a 2-factor model did a little bit better, but a 3-factor model really seemed to balance that relationship between fit and parsimony in terms of how we were representing the data. And what I would highlight here is that the way that the items kind of segregated across these factors actually does make a lot of post hoc theoretical sense. So you see on factor 1, these kind of mood, misery, and irritability items, maybe this kind of negative affect factor guilt, hurt, and embarrassment around maybe this kind of social reactivity factor, and nervous worry, tense and nerves, maybe this kind of more anxiety type factor. And while these factors are still highly correlated, they are somewhat dissociable. And in fact, when we run multivariate GWAS to look at SNP effects on these factors, we do find somewhat divergent biological pathways that we might miss if we were to employ an approach where, say, we just kind of sum across these items and run a GWAS on a sum score of the 12 neuroticism items. So I think this really highlights the ability of these sort of model fitting procedures to pull out some pretty interesting patterns in the data. Moving on to this new level of analysis, stratified genomic SEM, that we introduced in a paper that's currently on medRxiv and out for review right now, but the code for that is live on the GitHub along with a tutorial page for that. Is really designed to think about how we can start to make sense of GWAS findings characterized by thousands of genes that really individually explain only a very small portion of the phenotypic variance. So just as sort of a kind of hypothetical example, let's think about these future Manhattan plots where the GWAS sample sizes are getting in the millions, and now we're starting to see that basically the entire genome is somehow associated with the phenotype. Again, I know this is just sort of a case example, but it just highlights that, you know, at an individual SNP level, we're going to get to a point where the picture is so complicated that it's really hard to make sense of what's going on just based on a Manhattan plot. There's already a number of methods out there that in the univariate case can be used to basically partition heritability by using collateral gene expression data, such as what you might get from RNA sequencing methods, to try to lump associated genetic variants and portions of heritability into meaningful categories. So in this partitioned LD score regression paper from 2015, they showed that disorders like schizophrenia and bipolar disorder are enriched in the central nervous system, which you can see here based on these orange bars for that annotation, including for years of education as well, which of course makes a lot of sense. So we extend that same model to be able to look at partitioned coheritability. So this will look familiar to anyone who's worked with the bivariate LD score equation before. And really all we're doing now is that instead of using the LD scores here, we're using the partitioned LD scores. So the LD scores within a particular functional annotation. So we developed this and validate it as a means to an end for really being able to then feed these partitioned covariance matrices into Genomic SEM to then be able to examine genetic enrichment of any model parameter estimated in Genomic SEM for this kind of new extension that we're calling stratified Genomic SEM. So you can look at enrichment of residuals in a structural equation model, enrichment of correlations between factors, and I think what people would typically be interested in, enrichment of the factor variances to see where these kind of overarching factors that explain variance in the indicators are really enriched. So in that way, we can take a Manhattan plot of these common factors and start to look at these kind of top peaks and ask whether or not these hits are really enriched within genes that are expressed during certain developmental periods, such as during early kind of even prenatal periods or later in life, whether they're enriched in specific brain regions or even in certain neuronal subtypes. And this method really starts to, and you know, any kind of partitioning method starts to become increasingly exciting as the gene expression work starts to move at a rapid pace, and our ability to build these categories in meaningful ways starts to also really increase and become quite exciting. So again, this method is about asking whether there are certain biological functions that can characterize genetic variants with pleiotropic effects, which, you know, there's a lot of kind of convergence of findings across disorders that tend to cluster together. Unsurprisingly, psychiatric disorders tend to be enriched within brain regions, but now we're really trying to quantify that using this multivariate method for stratified analyses. And at the most fine-grained level of analysis, Genomic SEM can be used to perform multivariate GWAS to look at SNP effects on any model parameter that you would estimate in Genomic SEM. The way we do that is that we extend that S matrix I showed you earlier when walking through the two-stage process, where we've got that same genetic covariance matrix from LD score regression here in blue, and then we append this SNP column that include the SNP variants from a reference panel and the betas from the GWAS summary statistics scaled to covariances using that same SNP variants from the reference panel. And so what you would do is the package builds this matrix as many times as there are SNPs present across the indicators, and then runs any particular model that you might specify that includes an individual SNP. So for example, we look at SNP effects on a P factor that's defined by these 5 psychiatric indicators of schizophrenia, bipolar, major depression, PTSD, and anxiety. We take this LD score regression matrix and append the SNP column, and then we're able to look at the effects of an individual SNP, such as this particular SNP that we find is genome-wide significant with respect to its effect on the P factor. In the context of multivariate GWAS, we also have developed this SNP heterogeneity metric, which we call QSNP, because of its similarity to the meta-analytic Q statistic, that really ask whether or not the associations between a given SNP and the individual disorders, like I'm showing here in panel A, is sufficiently captured by a model that posits a single association between the SNP and the factor. So really, is it kind of, if you just fit this one relationship, is that obscuring the relationships that you might get if you fit all of these individual relationships? So we compare this common and independent pathways model, and if this model in panel A fits much better, then that suggests that this SNP is not really operating through the factor. And an instance where you might see that is if, for example, there's highly disorder-specific effects. So if a SNP really is specific to one of the indicators and has a null effect or an opposite effect on the other indicators, then you would expect to get a significant QSNP metric. And this really highlights that Genomic SEM is not about boosting power for the individual traits in the way that some other multivariate methods are designed, like MTAG, but is really about finding the SNPs that operate through the factors and the SNPs that are highly specific to the disorders, so we can start to get a sense of the multivariate picture across the different disorders that we include in the model. This is one particular application of a model that you could fit in Genomic SEM that people have shown some interest in, namely GWAS by subtraction, where you fit this sort of Koleski model for 2 traits, and then you have the SNP predict the 2 latent factors within the Koleski model. So in this particular example, we're looking at the effects of a SNP on cognitive performance and on educational attainment minus the genetic overlap with cognitive performance for what we've called this kind of non-cognitive element, so that then you can start to kind of break apart something that's really multifaceted like educational attainment and ask what SNPs underlie this overall genetic signal that are separate from the cognitive component. This is in a paper that is currently on bioRxiv but is forthcoming, and this is just to highlight that then you can produce these Manhattan plots for the cognitive phenotype and the non-cognitive phenotype down here, and identify these kind of dissociable genetic signals. And then in the paper, they also look at kind of polygenic score prediction for these 2 different phenotypes and find divergent patterns of prediction and also genetic correlations with outside traits. So this is just kind of one way that you can apply Genomic SEM in interesting ways. In general, it's just a kind of sales pitch for Genomic SEM. The different groups that have used it and using the open source R package have been fairly successful in publishing in a lot of different outlets. So this is just kind of a flavor of the different ways that people have been using it and the outlets that they have been publishing in using Genomic SEM. So with that, I want to transition as someone who's trained in clinical psychology to my main interest in psychiatric phenotypes and how I've used Genomic SEM to really understand the multivariate genomic architecture across psychiatric traits. So I just want to kind of start by explaining why I think multivariate work in psychiatric disorders is so important to begin with, even ignoring the genomic piece. So in this orange circle, I'm just showing kind of all individuals with mental disorders that will meet criteria for a disorder in their lifetime. And among that group of people, about 2/3 are going to meet criteria for a second disorder, half will meet criteria for a third disorder, and 41% will meet criteria for a fourth disorder. And while we know that our categories are not perfect, we do kind of think of them as sort of these mutually exclusive things, where I think when clinicians sometimes talk about people having multiple disorders, there's this sense that they've just kind of had bad luck in terms of maybe the environments or genetics risk factors that they've been exposed to. But really, at the end of the day, I think this speaks to how much our categories overlap pretty substantially, and how much we should think about kind of refining future versions of our diagnostic manuals so that we can create more mutually exclusive categories. And I think there's a lot of really compelling reasons to think about why we would want to do that. But before I get to that, this is just kind of depicting the extent to which we might kind of loosely think about these patterns of comorbidity and kind of the loose, rough borders that we've drawn between the disorders as being somewhat genetic in nature. So on the x-axis here, we have parent disorders across depression, generalized anxiety, panic, substance use, and antisocial personality disorder. And on the y-axis, the odds ratio for their child developing a particular disorder. And what we see is that the children are really at risk for any disorder, and not just the disorder present in the parent. Now, of course, that could just mean that a parent having a mental disorder is sort of generally stressful through an environmental component that kind of generally leads you at risk to any disorder, or it could mean that the genetics passed down from the parents are really kind of unspecific in terms of how they convey risk. But at the very least, it suggests that the boundaries between the disorders are pretty blurry at the end of the day. And so again, that's important because as someone who also currently practices, it can be very stressful to meet with someone and tell them that they've got every disorder in the book. I think this has real clinical implications in terms of the message that sends to the patient in the room, and also what it means for a treatment provider to think about what kind of intervention to give somebody when they're meeting criteria for multiple categories. And from a scientific perspective, I think this also has a lot of important ramifications, where if you have scientist A studying bipolar disorder, scientist B studying schizophrenia, and this fellow studying OCD, but at the end of the day, we know that these disorders are all related in some pretty substantial way. It probably isn't the best use of grant funding money to give all of these people a separate pool of money and then just kind of send them off in separate directions when they're really studying these phenomena that are actually interrelated in pretty substantial ways in some level. And so that is why, as a kind of backstory, I'm particularly interested in understanding the genomic architecture across different disorders, which I think doing it in a genomic space has a number of advantages. One, it can give us insight into what we know is a pretty important component of these disorders, since they're all estimated to be pretty heritable. But one limitation when people are understanding what's called as this general psychopathology factor or transdiagnostic P factor is family-based research on P is inherently limited to relatively common disorders, because it's going to be next to impossible to obtain genetically informed samples on rare disorders. And in just a basic cross-sectional sample, again, I noted that some of the disorders actually disallow one another. So it would be not possible to actually examine disorders within the same sample. So for the first time, methods like Genomic SEM, building off LD score regression, allow us to look at the genetic structure across both common and rare disorders, because LD score regression is able to estimate the genetic relationships across samples that are potentially independent at the end of the day. I want to walk through the different iterations of these factor models that we've done now. So I showed you this earlier in the context of the Snip model, but at the base model, we modeled in that initial paper the relationships across these 5 disorders and pulled out what we called at the time this kind of general psychopathology factor. I kind of am referring to it as a so-called general psychopathology factor because we also only had 5 disorders at the time that were sufficiently powered to put in the model. And as we started to include more disorders, this model started to look a little bit more nuanced than just a single common factor. So in the second major iteration of this, we worked with the cross-disorder group from the Psychiatric Genomics Consortium to publish this paper in Cell, where we looked at the genomic relationships across these 8 disorders of anorexia, OCD, Tourette's, schizophrenia, bipolar, MDD, ADHD, and autism. And what we found is that these kind of segregated into these 3 factors that we loosely call a compulsive disorders factor, a psychotic disorders factor, and a neurodevelopmental disorders factor. And I'll just briefly mention too that things like AN, OCD, and Tourette's loading onto the same factor is very consistent with what we might expect based on comorbidity rates and kind of convergent patterns across different research groups. Same thing for schizophrenia and bipolar. One kind of lone wolf here is MDD, and we were able to improve on that when we, in our most recent iteration of this model, now include 11 major disorders, now with alcohol use disorder here in red, PTSD and anxiety. And with the inclusion of PTSD and anxiety, we're now pulling this fourth internalizing disorders factor. But I would highlight also that for a number of these disorders, the sample size is updated pretty substantially relative to the previous iteration. And even with that update, we were still pulling out the same 3 factors when going through the exploratory factor analyses. So again, this kind of indicates that there is a lot of intercorrelations across these disorders, but there is some kind of nuance in terms of how these disorders kind of segregate into these subgroups. And in fact, if you just kind of look at the genetic correlation matrix across these 11 disorders, you can kind of see these factors kind of pop out when you order the matrix according to the factors that we modeled, where you've got this kind of subcluster up here, factor 1, this really tight cluster between schizophrenia, bipolar, and factor 2, and in particular, this really apparent cluster between PTSD and MDD and anxiety on that internalizing fourth factor. Because there were correlations across these factors, and because of the kind of growing interest in this overall P factor, we did model an overarching P factor that explained the genetic relationships between these 4 different factors, and found that this fit the data pretty well. But given these kind of partially competing models of either kind of 4 correlated factors or an overarching factor, we wanted to go on to do some other analyses to really evaluate the utility of each of these factors for understanding shared biology across their indicators. And we did that in a number of ways. One is we took the same logic that we developed for Q-SNP and applied it to examining the relationships between relevant external correlates for psychiatric disorders. And so again, in a very similar manner to Q-SNP, we ask whether or not the associations between an external trait and the individual disorder shown in panel A is sufficiently captured by a model that just shows a single relationship between the external trait and factor 1. And we compare the fit of these models and would expect there to be a really significant decrease in fit by just modeling this common pathways model when these associations are really discrepant across one another. We fit these sorts of models for 49 different external traits, and I just want to walk through a number of these findings. So I'm showing a couple different things here. Color-coded at the top are the 4 different factors from the correlated factors model, and then the P factor in turquoise. And then whether or not the bar is shown with a solid or dashed outline indicates whether or not it was significant for that Q trait metric. Where bars with a dashed outline we found to be significantly heterogeneous across the indicators. So within socioeconomic outcomes, we see that there's a relatively homogeneous relationship between a lot of these outcomes and the compulsive disorders factor in the positive direction. When we look at health and disease, we see a lot of positive correlations between the internalizing disorders factor and these health and disease outcomes, which is very consistent with phenotypic work. And within anthropomorphic traits, we do see, I think, a particularly interesting finding for the compulsive disorders factor of a negative correlation between body mass index and waist to hip ratio. And I mark that as interesting because anorexia loads on that factor. And you might think that that is really driving the relationship between BMI and waist to hip ratio, given low weight status as a diagnostic prerequisite for meeting criteria for anorexia. But in fact, we see that there are actually shared pathways with OCD and Tourette's between these external correlates, indicating that there's some sort of shared general risk between kind of these anthropomorphic traits and this compulsive disorders factor. And then finally, we see that there's a lot of homogeneous relationships with neuroticism, but not with this internalizing factor in neuroticism, which might come as some surprise, but is largely driven by a much higher genetic correlation with the MDD indicator. If we look at these correlations overall, we see that in particular, there's a significant Q trait metric for the neurodevelopmental and P factor, indicating that a lot of the relationships with external traits are not operating through these factors and suggesting somewhat limited clinical utility for these two factors relative to these compulsive psychotic and internalizing disorders factor. We then went on to perform multivariate GWAS in sort of two main steps. In the first one, we looked at the Snip effect as it concurrently predicted these four correlated factors, and then a Snip predicting this overarching general psychopathology factor. Just to orient you to these Miami plots, I'm showing on the top half the factor Snip effects, and on the bottom half the Q Snip effects that I've talked about earlier in this talk. In black triangles, I'm showing the 132 hits across these four factors that were NLD with hits that were identified for the individual disorders that defined the factors. In red, I'm showing 20 novel hits that were not significant for any of the individual disorders, which highlights the ability of Genomic SEM to make novel discovery even without collecting any new data. And again, I'll say that with the caveat that I really try to be careful to not frame Genomic SEM as a method for boosting power, but for ultimately looking at shared genetic signal and divergent genetic signal across different traits. But at the same time, because you are leveraging shared power, you are going to get some kind of boost in signal for a lot of these disorders. And then in purple, I'm showing the purple diamonds, the significant Q Snip effects. So for the compulsive disorders factor, these particular disorders were not super well powered, so there's not much signal. We see a lot of shared signal for the psychotic disorders factor. For the neurodevelopmental disorders factor, we see a sort of somewhat even balance of factor and Q Snip signal, and internalizing disorders factor, a much stronger signal for the factor. And if we look at the QQ plots for these different disorders, what I'm showing here in blue is the signal for the factor, and in pink, the signal for Q Snip. And what we would expect if the factor is generally capturing the relationship between the individual SNPs and the individual disorders that define the factor is that this blue line would sit kind of nicely above this pink line. And that is what we observe for the compulsive psychotic and internalizing disorders factor. But on the other end, we see that the QSNP metric is actually much stronger than the factor signal for the neurodevelopmental disorders factor, which indicates that a lot of the genetic signal is not operating through the factor. So similar to what we observed for the patterns with external traits, for individual SNPs, we find that they are often not operating through that neurodevelopmental factor. And if we look at the individual SNP effects for some of the SNPs that were particularly SNP significant, we see that a lot of that is due to divergent effects for autism, which also loads on that factor and may come as no surprise. So that's not to say that a neurodevelopmental factor in a kind of hypothetical sense might not prove to be useful, but at least in the way that we've defined it, based on the data that we had, it doesn't seem to be sufficiently kind of explaining the shared signal across its disorders. And then if we look at the signal for the P factor, we see a really striking imbalance between the factor signal and the Q-SNP signal, where we see only one hit that was an LD with an individual disorder hit, no new loci, and 69 Q-SNP hits, and just a really elevated pink line relative to that blue line, indicating that almost none of the SNP effects are operating through that P factor, which really suggests that there is kind of limited utility to a P factor for actually understanding shared biology across disorders. Finally, I just want to kind of go over the stratified genomic SEM findings for this particular 11 disorders model, and I want to focus on some findings for enrichment of brain cell types. So we know that psychiatric disorders are generally enriched for genes expressed in the brain, which is sort of, I feel like more of a sanity check to see that it's not expressed in, the spleen or the stomach, but is really in the central nervous system like we'd expect. But with more recent single cell sequencing efforts coming out, people have been able to pair that up with GWAS summary data to actually look at enrichment of specific brain cell subtypes, which gives us maybe a little bit better target in terms of where that signal is coming from within the brain. We also know that protein truncating variant intolerant genes which is referring to specific genes that generally do not show mutations, are enriched across disorders. So these genes might be particularly relevant for just kind of functioning in general. And so what we did is we created annotations for PI genes, brain cell subtypes, and their intersection to examine whether or not prior enrichment patterns actually reflect some sort of pleiotropic signal across disorders. So here I'm showing those stratified genomic SEM findings for examining enrichment of the factors. In orange, I'm showing the glial cell category. In dark blue / purple, the excitatory brain cell subtypes, and then the GABAergic subtypes, the PI genes, and then their intersection over here in the rightmost part of the plot for the different factors. And what we see is that there's a really unique signal for the psychotic disorders factor within excitatory and GABAergic genes within the PI genes, and a signal that is particularly enriched for their intersection. And so I think this starts to give us some real traction in terms of being able to understand what it is that this psychotic disorders factor is capturing in terms of where that genetic signal is coming from, and also starts to point towards some reasons that these disorders might actually diverge and actually look both phenotypically different and not share genetic signal at the level of this P factor over here. And I also would just highlight that if you were to go into Google Scholar right now and type in any of these neuronal subcell types for bipolar disorder, schizophrenia, you could really make a case based on the prior literature that really any of these are relevant to the factor. And of course, this is just one study among many, but we're using a pretty high-powered set of GWAS summary statistics paired with some pretty cutting-edge RNA-seq data that shows that really these glial cells are not particularly relevant for the psychotic disorders factor as we've defined it here. So in conclusion, we've really expanded the genetic factor model of psychiatric risk to identify 4 major factors of genetic risk sharing. We find relatively high utility of the psychotic and internalizing disorders factor when we go on to really kind of stress test these factors using different follow-up analyses, including those Q-SNP and Q-trait findings I talked about earlier, where we find that the associations between relevant external traits and individual SNPs are generally captured by the factor. For the compulsive disorders factor, I would really frame this as a factor where the jury is still out in terms of its utility, namely because the GWAS summary statistics that define that factor are still relatively low powered. We do see that it does a pretty good job of explaining relationships with external traits, but at the level of individual SNPs, there's just not really the signal there to see whether or not it's operating through the factor or through the individual disorders. And for the neurodevelopmental factor, we really see some pretty low utility where a lot of the relationships in the disorders that define this factor are relatively unique in terms of their patterns of genetic correlations with external traits, and a lot of the SNP associations did not operate through the factor in a way that seemed largely attributable to a signal that was unique to autism relative to PTSD and ADHD that also loaded on this factor. We also know that we can model a P factor using a genetic correlation matrix in line with a lot of phenotypic and family-based work that's been done. But when we stress test this factor, we find that this in particular has incredibly low utility to the extent that it obscures relationships with external correlates and SNP associations. This might be due to the fact that there are sort of kind of unique bivariate associations between different factors that are not captured across the factors as a whole. So for example, there might be some sort of shared signal between the psychotic disorders factor and compulsive disorders factor that results in that genetic correlation between those two factors that is really dissociable from the signal between a psychotic disorders factor and an internalizing disorders factor. So it's kind of this complex Venn diagram across these factors that does not include this kind of P factor at the center, at least in the way that we've examined it here, which we think has pretty broad implications for a pretty rapidly expanding P factor literature with a lot of articles coming out all the time about this P factor. Using stratified genomic SEM, which is that new Genomic SEM edition that is live on our GitHub. We find that prior enrichment findings generally reflect broad pathways of risk. And we find in particular that the intersection of PI, excitatory, and GABAergic genes are enriched for this psychotic disorders factor, which gives some real insight into the biological pathways that might underlie the really high genetic correlation between these two very debilitating disorders of bipolar disorder and schizophrenia. And again, as RNA-seq methods get even better and the corresponding univariate GWAS become even better powered, we're going to be able to make these categories even more refined and even put them within developmentally specific windows, such as excitatory neurons expressed during a specific period of development, which I think is, just at face value, could be a really exciting set of findings. And just to end on a kind of sales pitch note, a lot of you have heard me talk about Genomic SEM, but I hope that it's clear that this is a pretty flexible method in terms of its ability to ask a number of different interesting questions, whether you're looking at something like GWAS by subtraction, just doing some general factor modeling, or trying to examine systems with relationships across traits that you might not be able to otherwise examine because of how rare or mutually exclusive they are. It is an open source R package that's publicly available with a GitHub and a Google group that you can ask questions on. And Stratified Genomic SEM is now live on the GitHub. And one of the reasons that it's exciting to talk to a group like this is not just to talk about the work that I'm doing, which in and of itself is fun to do, but it's also great to hear what kind of questions people have and also potentially develop collaborations, projects, or grant ideas. And just as an aside, I'm on my internship, my clinical internship year at Mass General Hospital in Boston, but I will be starting as an assistant professor at CU Boulder in the fall. So again, if people have projects or grants that they want to work on, this is obviously a group that I would be particularly interested in collaborating with. And so I'll just end by naming and thanking a number of different people, in particular Elliot Tucker-Draub and Michelle Navard, who have been really central to working with me to develop Genomic SEM and its extensions, and a number of different people named here, and of course also thanking groups like the Psychiatric Genomics Consortium, IPSYCH, and UK Biobank that really contributed to the data sets that I presented here in terms of the application of Genomic SEM to psychiatric traits. So that's all I have. But again, I just want to thank everyone for inviting me and for your time, and curious to hear what questions people may have.
Andrew, thank you for a fascinating talk. How I'd like to structure it, the Q&A, is that Sam Trejo, who suggested you come talk to us, and we really appreciate that suggestion, will give us the first question. And for those who have follow-up questions in that conversation, I suggest you jump right in. But for those who have other questions, if you could just queue up in the chat, I'll moderate that to tell us who's next. So Sam's going to give us the first question, and then we'll go from there.
Sounds great.
Hey, Andrew, really cool talk.
Hi, Sam. Thank you.
My question is kind of about this idea that I think is true for this earlier stuff you talked about with the GWAS by subtraction and the sort of non-cognitive, cognitive parts of educational attainment, but it seems like it extends later on into the stuff that you're doing with like the P factor and all of these different sort of kind of like psychiatric factors. So the first question is, like, with the GWAS by subtraction, if I were just to take the linear difference between the EA sumstat, like, beta weights and the IQ beta weights, I would do like a less sophisticated version of, I think, what you guys do in that paper, right? Like, I'm just sort of taking the bits in one GWAS that aren't in the other GWAS. And I was kind of curious, like, how similar of an answer would I get, do you think, or do you know, to, what the Genomic SEM model fits? And then what are the advantages using Genomic SEM? And then my kind of second question that's related to all this is, like, well, actually, I'll just let you answer that one first.
Yeah, no, that's a great question. So Michelle Navard actually has an alternative method that people are probably aware of, GWIS, Genome-Wide Inferred Summary Statistics, that does something very similar to what you're describing, that just kind of takes the summary statistics and at that level just kind of pulls out the shared and unique genetic signal. I think the advantage of Genomic SEM is that is two things. One, that you can kind of actually depict the relationship between these two different sets of traits within like a classic Koleski model in a way that's sort of intuitive in terms of how the genetic relationships are shared across these different traits that you're including. And the other is that my sense is that if you do just kind of what you're describing, you're potentially going to get biased results because of that sample overlap piece that Genomic SEM is able to account for using the V matrix. So if you know that you have two entirely independent samples and you have no concern about that, then I don't know, I think it's kind of an open question whether or not the answer would be pretty similar, but my sense is that people are generally pretty concerned about some level of unknown sample overlap, and Genomic SEM is going to give you some ease of mind in that case that you're actually appropriately accounting for that.
Yeah, that's helpful. That's helpful. And so then kind of the next question is like, in both cases, I think with the cognitive and non-cognitive and then all these psychiatric factors, like we're now able to basically, you know, isolate and generate polygenic scores for traits that just don't exist. And I guess, I mean, by that, I mean, they don't exist as like they've never been measured, and it's not clear that we would ever be able to measure them. And I was just curious if you know, what you thought about that and whether those sort of polygenic scores, should we, like, you know, we should use them differently or think about them differently?
Yeah, that's another great question. I mean, I think about that too in terms of the factors that we pull out too. I mean, this reflects some sort of shared genetic signal across these disorders that isn't actually directly observed. And so I think it's always really important to kind of do some follow-up just for yourself and for the people who are reading this kind of hypothetical article that you're putting together to actually characterize what that kind of new polygenic score looks like. So for example, by taking non-cognitive summary statistics and looking at the genetic correlation between a bunch of external correlates, so you get a sense of what that signal is actually picking up on in a sort of kind of multi-dimensional space. So I totally agree. I don't think we should just start like kind of removing things from one another and just start kind of kitchen sinking things without any kind of hypothetical guiding force or sense of what these new summary statistics are picking up on at the end of the day. But I do think there's a number of things within Genomic SEM that you can do to kind of clarify that.
Thanks, super helpful.
Yeah, great
you have a
question.
question.
Oh, yes. Thank you for the talk. It's really comprehensive, pretty much covered everything about Genomic SEM. So I have questions about some technical details, but for the follow-ups, probably you can continue in our individual session after this. But for now, I'm very interested in the annotation stratified Genomic SEM. So I'm wondering, because if you're interested in the heritability enrichment of factors in certain regions, you actually don't have to fit annotation stratified version of Genomic SEM, right? You can just run the standard Genomic SEM and get the GWAS summary set for those factors, and then test if their heritability is enriched in certain annotation categories. I wonder empirically, do you actually see differences when you run the annotation stratified version of GSEM and then characterize the annotation enrichment?
You do see some differences. In general, you just see kind of deflation of signal. I think that's a great question. You know, I think that's one thing that comes up a lot is just this general idea of like, couldn't you do this in a much simpler, kind of more straightforward way by just taking the summary statistics for the factors and running partitioned heritability on that. The reason that you see deflated signal is that the factor summary statistics are estimated with error. And what I mean by that is those summary statistics are going to include some of that Q signal or signal that is not actually operating through the factor itself. So you could think about kind of pruning based on significant Q SNPs and then feeding those summary statistics into partitioned heritability. But I think it's kind of unclear what threshold to use when you do that. Whereas if you're doing it strictly within a genomic SEM framework by looking at enrichment using partitioned covariance matrices, you're not including that kind of error that gets introduced into the summary statistics in that way. Another thing I would say is that you can also look at enrichment of the uniquenesses and enrichment of things like factor correlations. So that is sort of a tangent to the question that you're asking, but just to say that stratified genomic SEM, I think, is kind of more broadly useful in the sense that you can look at things that you couldn't really feed into a partitioned heritability analysis, like enrichment of factor correlations, or kind of concurrently looking at enrichment of the uniquenesses within the same model.
Yeah, this is very helpful. So a quick follow-up would be that, do you have a sense about how big the annotation need to be for this to work? Because my intuition would be that if you have a very small function annotation, the annotation stratified genetic covariance estimate will become very noisy. So then when you fit a separate genomic SEM in that genomic region alone, maybe it will be challenging to converge, right? So empirically, based on the sample size in your analyzed summary sets, how big can the annotation be?
One thing as far as the model converging is that the way that the estimation procedure is coded is that we kind of fix the estimates from the annotation that includes all SNPs and then re-estimate the model with those fixed estimates in the partition-specific covariance matrices. So that helps a little bit with just kind of wonky model estimation. In terms of just like genetic covariance estimates that are really imprecise, I know that this is clear to you, but just to sort of put out there that, we have the partitioned sampling covariance matrices too. So you don't run a danger of false positives in the sense that if a covariance estimate in the partition space is really imprecise, then it's also going to have a huge standard error that ports over to the enrichment estimate. So there are certain annotations that are really small where you get like a huge enrichment estimate, but with a confidence interval around it that is humongous at the same time. So big point estimate, but not significant in that sense. In terms of annotation size, I don't have like a great sense of how small it needs to be. I mean, these PI by brain cell subtype annotations are not humongous. Yeah, I don't have a concrete answer for that. The other thing I'll say is that when we first started this project, my interest was in actually looking at whether or not the factor structure changes across annotations. And that we are not powered to do for the reason that you're kind of highlighting, that there's a lot of just kind of random noise in the covariance estimates so that you get these kind of fluctuating factor structures that don't actually reflect something that seems to actually be changing in the population. So that's why we're not doing kind of partition-specific factor structures, but more kind of fixing the model in the genome-wide sense and then looking at enrichment of particular model parameters.
Yes, great, thank you.
Of course.
We had an overlapping question that Philip and James asked about model fit. I think the general question is how you think about model fit, but maybe James could fill in a more precise, targeted question.
Sure, yeah. Thanks for the talk, Andrew. That was extremely helpful and informative and exciting. So my question, I think it's similar to Philip's, although he can certainly jump in here, is that when we've tried to fit these more complicated models in GSEM, where we have more than just the 5, right? Like you did with the 11, the problem that we ran into was that the more complicated your model gets, the harder it is to actually produce a strong fitting model. So I'm just wondering, since you didn't really talk about it in your slide, is when you were able to successfully extract these 4 different latent factors, how well would you say that the overall model actually fit the data to the extent that you feel confident that this neurodevelopmental factor is in fact a truly unique factor? Can you speak a little bit about that?
Yeah, so that 4-factor model does fit the data well by conventional standards. So CFI, I think, is like 0.96. SRMR is, I think, 0.06 for that model. So using like these kind of arbitrary cutoffs, we find that the model fits pretty well. We see really clear increase in model fit as we kind of move from a common factor model to this more nuanced 4-factor model. Of course, there's the trade-off of, a more complicated factor model is always going to fit the data better. But it kind of hit this point where hypothetically the factors that we were pulling out made a lot of sense. It seemed to fit the data pretty well. And as we kind of included, updated summary statistics, we were consistently pulling out the same factors using a kind of restarted exploratory procedure. So at the same time, I think that you can have genetic correlations are such a broad kind of 10,000-foot metric that in the way that we showed with these kind of Q follow-up analyses, it's important to really stress test these factors because, we can model a P factor, we can model a neurodevelopmental factor, but that might be that it's kind of aggregating across these really dissociable pathways at different levels of analysis. So
Yeah,
I think that model fit is just one piece of this puzzle.
I agree entirely, and I'll follow up with you in our individual meeting. But one additional thing I just wanted to add was the alignment of the models that you extracted and with theoretical alignment with our theory of what these factors should be. So, for instance, in your neurodevelopmental factors disorders, you had PTSD, autism, ADHD, and I think Tourette's, right? So, to what extent does that align with, for instance, high-top models of neurodevelopmental disorders or even DSM perspectives of what neurodevelopmental disorders are classified as? Not that we necessarily agree with the DSM, but I'm just saying that, there's the quantitative piece in which the disorders fit this type of structure, but then there's also how well does that fit with our theoretical understanding of how these disorders should look or how they present?
Yeah, I mean, it's sort of a mix of both. And I think that that's, in some ways kind of nice. Like you would hope that after spending millions of dollars to do genomics, we don't just recapitulate what we already knew in some way, but actually get some kind of novel insight. And the real interesting thing within this neurodevelopmental factor is PTSD. And the reason I highlight that is we actually see that PTSD and ADHD are correlated greater than 0.7 across multiple separate cohorts, which is not something that you would get from reading the DSM or just from practicing clinically, I don't think. And so there's sort of a separate project going on that's really trying to tease that apart. ADHD and autism, I would argue, there's some good reason to think they would load together. But in a purely kind of exploratory sense, that particular factor, I think, is the odd one out, particularly because PTSD is loading so strongly due to its relationship with ADHD. But that relationship is there, and we feel pretty confident that it's there because of how consistently we see that across independent cohorts. Yeah.
Philip, is one of your questions short enough that it would be a minute or two long?
Oh my God. I'm not really sure, but anyway, so let me just briefly say, Andrew, I'm so glad that I heard that you accepted this job offer from Boulder. So I know that Matt Keller is like super excited about, that you're coming there, and I only heard it from him like very recently that this is finally working out. So thumbs up, congratulations.
Thank you.
So actually, I had a lot of like very far drifting like comments and questions that may actually be really better if I talk to you separately about this. But maybe just one very concrete thing. So I was just wondering if Genomic SEM actually makes use of the standard errors that LD score regression RG estimates spit out, or are you only using the point estimates themselves?
We're also using the standard errors in that V matrix.
Okay, well, so then there is this slightly weird thing that if you construct the genetic correlation matrix with a bivariate method that doesn't take the entire multivariate structure into account, like LDSC, right? So that's really just bivariate. Then the resulting matrix may actually not be a positive semi-definite, and the standard errors are actually not necessarily theoretically correct. So I mean, there is a way how you can actually get the correct standard errors, but it would be a completely different method. It would basically be a multivariate version of Gremel, which would have some advantages, some disadvantages. But I'm just thinking, is it theoretically possible that you fit that you build your genomic SEM model based on standard errors and RG estimates that are not coming from LDSC, but let's say from multivariate Gremel?
It is, and I think at one point Ronald was maybe working on that even.
Exactly. Yes.
Yeah.
So this is where this is coming from. Exactly. Yeah.
Yeah. And there's it's sort of like this kind of constant thing that is like, you know, these different kind of bivariate methods come out to how we could maybe incorporate that to kind of build out these matrices. I will say that we, you know, the sampling covariance matrix is estimated within our multivariable version of LDSC, and the block jackknife kind of proceeds in a way that is similar but different in that we're also populating those off-diagonal elements, but that actually doesn't answer your question. So your point still holds. And I think, yeah, trying to incorporate some of the stuff that Ronald's been working on is a real interest.
Cool.
Yeah.
All right, thanks.
Of course.
I think we could keep you for a long time with these questions and answers, Andrew, but we thank you again for coming. I know you're going to stick around for the beginning of your individual meetings.
Yeah.
So I want to just, on behalf of the group, thanks for such a clear and interesting discussion and presentation.
Yeah, thanks everyone. Really great questions, and it was a real privilege to present to you all.